All right, welcome everyone, and thanks for joining today as part of the Soil Health Training Program webinar series. Welcome to Webinar 2, Soil Structure and Chemistry. Once again, thanks to the Lake Champlain Basin Program and NUEPIC for funding to create and host this Soil Health Training Program. Couldn't have done it without you. The agenda for today, I'm going to do a quick course overview just to figure out where we are, a reminder to sign up for campus, a slight homework review, and then we're going to turn it over to our presenter, Fred Magdoff. And there will be 4 parts of his presentation today with Q&A in between. So thanks for staying engaged throughout, and then we'll go over the plan for next week. As a reminder for the course overview, we are in the middle of the 12 webinars, and so we've had one webinar last week. If you missed it, there is a rough recording on Zoom that you are able to access on the campus website. And then today is soil structure and chemistry, and you can see the other courses for the rest of the series on this page. Just as a reminder, everything that we are doing as part of this course is being posted on campus. If you have not yet set up your account, we do strongly encourage that you do. It'll give you access to all of the resources. The instructions are on the screen, and they should also be in a couple of emails from me, but please do reach out if you have any questions at all. Then I just wanted to do a quick little homework review. I do hope folks were able to read chapters 2 and 5 of Fred's book. On the screen is just a little bit of an overview of the first chapter of chapter 2. And what I would invite folks to do is to put into the chat any questions or things that they learned from the 2 chapters. Fred will be addressing many of the things that were in those texts today, but if you have any specific questions that you really want to make sure get answered, we have the expert with us today. So please do post in the chat if you have anything that you want to make sure gets addressed. And these are the topics from chapter 5 as well. All right, I'm going to stop sharing. And so our presenter today is Fred Magdoff, who is an emeritus professor of plant and soil science at the University of Vermont. His his interests range from soil science to agriculture and food to the environment to the U.S. economy. His research at UVM was on ecologically sound ways to improve soil fertility, especially focusing on the critical role of soil organic matter. He is the co-author of Building Crops for Better Soils, which you are all familiar with, and What Every Environmentalist Needs to Know About Capitalism, as well as a number of other books and technical articles on agriculture and on the U.S. economy. He has also written numerous articles on environmental issues, including ecological agriculture, production and use of biofuels, ecological civilization, population and global resource depletion, and the environmental and social problems of capitalist agriculture. So, thank you all for being here today, and with that, I will turn it over to Fred to go ahead and introduce yourself and take it away. Thanks.
Okay, so I'm on, huh? Okay. Well, so I do have the PowerPoint presentation, and I thought we'd go back and forth a little bit on it just to break it up a bit. Also, you know, I do have a significant amount of material on soil organic matter, and I see that you're going to have next week a whole session on soil biology, which I've included in it. So maybe I'll move through that a little more quickly than I would have otherwise. But so let me try sharing the screen, which should work. Let me know if it's not. Start share. Is this working?
That looks great, Fred.
Okay. Well, you'll see that again, but this is this is a picture which is actually on the back of the book. It's building soils for better crops. It's not building crops for better soils. By the way, Lauren, no criticism intended, of course.
My apologies.
Oh, it's, believe me, you're not the first one. Anyhow, I think most people can tell the difference between the soil on your right and the soil on the left, just with regard to the color, the texture, the amount of roots. You can see aggregates on the right. You see it more compact on the left. There's a difference between an unhealthy soil and a healthy soil, and you can smell the difference sometimes also as well. It's quite significant. So what I thought we'd start with, if I can get this to advance, is this, yes, it is. Okay. Okay, soil health is a concept, and then soil organic matter and soil health, how it relates to soil health is the second part of this, and then we'll break for questions if there are, and if there aren't, we'll just go on to the next part. So the thing that gives soil health to me its importance as a concept is that it's really approaching soils and plants, growing plants, agriculture, from a preventive approach. You're trying to prevent problems from happening. You're trying to design a system, an agro-ecosystem in which you will have fewer problems than you would in the traditional or conventional agricultural ecosystem, in which they use a reactive approach. That is, oh, we don't have enough nitrogen fertilizer. Well, we better go buy some nitrogen fertilizer and put it on. Soil's too compact. Well, we'll come in and break it up with a subsoiler, or there's solutions for everything. But you view them as individual problems. You have a pest outbreak. Well, you get this particular pesticide and you spray the crop, or you put pesticide on the seeds or whatever. So that's a reactive approach. So a preventive approach is just like in preventive medicine, same idea, is it doesn't mean that people won't get sick. It doesn't mean your agroecosystem won't get sick or your plants, but you're doing everything you can to create a healthy ecosystem. And you do that mostly by mimicking healthy natural systems. And that's the main approach. So what do I mean by a healthy soil? Okay. First, you've got sufficient nutrients for the crops that you're going to be growing, but not excess. That might cause pollution. Well, they do cause pollution if they're in excess, especially phosphorus and nitrogen. Good tilth, that is good soil structure. And we'll talk about that a little bit. Sufficient depth, of course, the soil can be great, but it's only 3 inches deep. And then you've got bedrock underneath it. That's not particularly useful soil for most agricultural purposes. Of course, you can come in and do stuff if you want to spend enough money and time and effort. Good water storage and drainage, and we'll come back to that. You have to be able to have water infiltrate into the soil and store there, be able to be stored there for plants to use, and you want the soil to be able to drain so it's not, all the pores aren't filled with water. Free of chemicals that might harm plants, and these could be from human endeavors or natural, like aluminum at low pH. Aluminum becomes very soluble, and you may have aluminum toxicity. You have to have a pretty low pH for that to happen. Pretty acid soil, that is. Low populations of disease and parasitic organisms, plant disease and parasitic organisms. High populations of organisms that help plants and help plant growth. And I presume that this will be gone into next week. I'm just doing this very briefly. I'll give a couple of examples, but there are a lot of organisms in the soil that actually promote plant growth. They either antagonize potential disease organisms or nematodes that might feed on plant roots, or they actually produce chemicals that stimulate the plants and provide the plants with self-defense capabilities. Plants are able to defend themselves. If you get to, I think it's chapter 8, there's a whole story about that in Building Soils for Better Crops, about the interaction between soils, plants, and their pests. Low weed seed pressure. Obviously, you'd like not to have a lot of weed seeds in the soil. And you like a soil that is relatively, well, resistant to being degraded. You can degrade any soil if you work at it. You can have the best soil in the world. If you want to beat the hell out of it, you can do it. Okay. You can go out there when it's wet, you can compact it. But soils that can resist degradation are obviously to be preferred, and you're trying to build that resistance to degradation into the soil. And then if a soil does get degraded by something or other, it has resilience and it can bounce back. So these are the characteristics that we'd like to have in a healthy soil. This is what we're aiming for in soil health. And so it's really a holistic point of view. It takes into account the physical characteristics of a soil, how it's put together, aggregation, the chemical characteristics of the soil, the pH, the nutrient availability. We'll get into cation exchange capacity, ability to hold on to nutrients, and the biological. So these are all integrated into the concept of plant health. We're not leaving any of those out. In 1908, there were three researchers at the University of Vermont who were tasked with trying to find out why the soils in Vermont had become run down. Okay, they were used up. Why are soils which in our father's hands were productive now relatively impoverished? This is what the Experiment Station Bulletin was trying to answer, that question. And it's a long conversation in the booklet, and they have some data and stuff. But this is their conclusion. The depletion of the soil humus supply, and humus is really a synonym for soil organic matter in those days, is apt to be a fundamental cause of lowered crop yields. Okay, this is 1908, so we're talking about 120, well, not quite, but we're pushing well over 115 years ago. And soil organic matter is at the heart of soil health. It's not the whole story, but it's an important part of the story. And that's important to keep in mind. And this is something which this understanding is relatively new. Now, when I went to school, that was a few years ago, of course, soil organic matter was mentioned in the textbook, and it was in the soil, but okay, so it's there. It's something that's there, but no big deal was made of it whatsoever. And it took me a long time to come to the understanding that not only is organic matter important, but it's at the heart of the story. Again, it's not the whole story, but it's a critical part of the story. And if you read those chapters, you'll see, and these terms come from those three Ag Experiment Station scientists. They said there are three different types of organic matter. There's the living, well, we'll talk about that. There's the dead, and we'll talk about that. And then there's the very dead, and we'll get into that. And when you talk to farmers, they like that. They like that that way of phrasing it, and I enjoy it too. So living organisms in the soil, there's all sorts of organisms, many different types of bacteria and fungi and nematodes, earthworms. You've got larger ones like moles, you've got mites, springtails, which are and plant roots. We shouldn't forget that. That's living in the soil, and they're organic. So it's part of the soil organic matter, are living roots. So anything that's living in the soil would really be considered part of soil organic matter. This is the one video that works, the least one that I wanted it to work, but it just shows you some aggregates. It's from the video Life in the Soil. If Lauren hasn't sent that out to you, it's a video worth seeing, made maybe 30 years ago in Japan, but a lot of photo micrographs and time-lapse photography. In this case, it's just moving, but some of them show the roots elongating and the root hairs coming out. It's really beautiful to watch if you're into that type of thing. These are amoeba getting ready to engulf bacteria. You can see them doing it on the video. Paramecium going around trying to find some bacteria to eat. This was this is a photograph that's in the book, a nematode this is the nematode here, that is feeding on the hyphae of a fungus. And so there are nematodes that specialize on feeding on fungi. There are nematodes that specialize at feeding on bacteria or other nematodes, or there are nematodes that feed on plant roots. And those would be considered to be plant pests. Obviously, we'd like to see those in very small numbers. And what's interesting is also there are fungi that feed on nematodes. And this is these are two photographs from that video. The one on the left showing the rings of this fungus, which can trap nematodes. A nematode swims into one of these rings, and the ring clamps shut. And the nematode, you can see on the video, the nematode on the right now, thrashes up, trying to get away and trying up and down. And it's finally killed by the fungus, and the fungus basically uses the nutrients in the nematode to live. So that's you've got earthworms, you've got all sorts of things crawling around in the soil. This is a picture of a root. I think it's in the building soils. Wheat root that's heavily infested with mycorrhizal fungi, and you can see the hyphae coming out and some of the small spores also of the fungus. And this is a beneficial fungus. It infects most of crop roots, I think, aside from the brassicas. I think all of the others it does. And it's like an extension of the root system. It helps plants get water and nutrients, and protects against organisms, mainly by, I think, physical protection of the sites. So mycorrhizal fungi are quite to be praised. So this is just from the book. Also, it's the food web in soils, and everything starts from the lower left, which is the dead. By the way, this is the dead, which we'll get to in a second. That is residues of corn. You can see there. It could be wheat or leaves or whatever. Dead insects or dead worms or whatever is there. And that becomes the basis then for everything that's living in the soil. And of course, this is all driven, I think, as you're all aware, by photosynthesis. It's all plant-derived energy from the sun. That's how we all live on Earth. And that's driving this food web through the production of plants or animals that eat the plants, and then they're recycling their residues. Okay, so the dead then are recently dead organisms. Crop residues provide the food for soil organisms to live and function. Sometimes referred to in the literature as active organic matter or particulate organic matter. Here's just some of the particles that you can see that we extracted some years back from soils here in Vermont. And then the very dead is not available for organisms to readily use. So this could be inside aggregates where organisms can't get to it. It's sort of tied up inside an aggregate. Could be absorbed on clay particles, very small organic molecules, or they could be very well decomposed organic materials that are not easily decomposable anymore. So that's the dead and the very dead, and all three types play an important role in helping to produce high yields of healthy crops. This is also from the book, but it's showing what happens when you add organic matter to a soil. Green manures, crop residues, etc. You increase biological diversity and activity. That reduces soil-borne diseases and parasitic nematodes. You have, through decomposition, aggregation is increased. Sticky substances are produced during the decomposition process. Pore structure is improved, which improves tilth and water storage. I'll show you some stuff on that in a sec. Humus and other growth-promoting substances are produced. Nutrients are released, and harmful substances can be detoxified. So that organic matter, through the process of being used in soils, has a tremendous effects. As a matter of fact, it's hard to find a soil property that is not affected by soil organic matter. There's only one that I can think of, and that is the texture. Soil texture is a name given to the amount of sand, silt, and clay in the soil. Well, that doesn't change. You can put a lot of organic matter in there, but that doesn't change the percentage of the particles of minerals that are in the soil, except as a percentage of the whole, because organic matter is, but if you leave out organic matter, those relative amounts obviously stay the same. But if you look at any other soil property, I'm pretty convinced it will be affected one way or another by organic matter management. So it's how you manage the organic matter, which is really the key question. Okay, what I'm going to do is just stop sharing here, and assuming that I can put it back on again relatively easily. And I don't know if you can see me better than you could before, but if there are any questions that you have on what I've said, I know I've gone through this relatively quickly, but about the living, the dead, the very dead, about the importance of soil. I presume you all know that you are made of soil, right? From soil thou art, and unto soil thou shalt return. It's not dirt. It's a mistranslation in the King James Version, okay?
Thanks, Fred.
Yeah, everything in our bones, our teeth, our everything in us has come from the soil and has gone through plants, and then we've eaten the plants, or we've eaten the animals that ate the plants. So you literally come from the soil, literally. Yeah.
Thanks, Fred. Kat, I see your hand is up if you'd like to ask your question.
Yes. Hi, Fred. So delightful to have you here talking with us. And in terms of very dead, I wonder if you could talk to us about fossil fuels. I think of that as being the ultimate in very dead.
Well, yeah, I mean, it's a little bit of a digression from this topic, but yes, fossil fuels are definitely quite dead. On the other hand, I mean, they are stored solar energy, just like the organic matter in the soil is stored solar energy. In us, I have stored solar energy in me. Well, it's going to run out if I don't replenish it. The problem with the oil, of course, is it will run out, but before that, we're going to cause a disaster for the earth. It's I sometimes laugh or chuckle when things seem so absurd to me, but that doesn't mean I don't think they're serious. And of course, global warming is terribly serious. And how we manage our soil, by the way, of course, I don't know if people are aware of this, but there's about 3 times more carbon stored in soils than in the atmosphere above us. Okay, so in all of the atmosphere above the earth, there's 3 times the amount of carbon stored in soils as soil organic matter than is in the atmosphere. And of course, as we work a soil, when you till a soil, by the way, when you open the soil, you break up aggregates, you leave it open for more rapid decomposition by organisms, and that causes more CO2 evolution. So one of the things that we're not covering today, but you're going to cover later on in this, is reducing tillage. And one of the things that reducing tillage does is it slows down the breakdown of organic matter in the soil. And so one of the reasons that we have so much CO2 in the atmosphere, the main one, of course, is the burning of fossil fuel. But one of them is the decomposition of so much organic matter from soils through agricultural practices, which depleted it. Maybe half the organic matter in some of our Midwestern soils, 40 to 50% has been lost. Where did it end up? Well, most of it in the atmosphere. So a lot of CO2 has gone into the atmosphere. That was a long digression from the question. I do that.
You're doing great, Fred. Thank you very much. There's not another question in the chat. One of the questions from last week that had come up was why, I guess, the very dead soil organic matter is less accessible to microbes than the others. And I'm not sure if that's something you'll talk about in the rest of the presentation.
Well, no, I wasn't going to go into it in any more detail. Basically, it's physically sequestered. In other words, it's inside an aggregate, which is so tight that it doesn't have enough pore space to allow in organisms that can access it, decompose it. Now you break up that aggregate, let's crush it. Oh, that's a whole other story. That's one of the reasons that tillage enhances the degradation of organic matter. So there's that type of physical, sequestration, if you will. There's also a physical connection that occurs with small molecules of small organic molecules, excuse me, that can be adsorbed, that is attached to clay particles, maybe some silt ones too. And those are not available. They're just physically aren't available to organisms to use. And then there's a whole debate about humus, about what is this strongly decomposed organic material. And it is apparently not as important as it was once thought. There's less of it in the soil as it was once thought. That some of it is really, that we thought was humus, is really an artifact of the chemical procedures that were used. But the basic idea is that either chemically or physically, the material is not available to organisms. Doesn't mean it will never be available, but it isn't available now.
Noted. Thank you very much.
Hey,
Yeah,
Lauren.
Willie. Yeah,
Hi. Yeah, thanks. I don't have a raise your hand button on here, so sorry
sure.
to butt in. Hi, Fred Magdoff. Good to see you. Long time no see, my friend.
Good to see you.
Fred, carbon cycling and carbon sequestering. Guess I'm kind of working from a supposition, I guess, to a point that increased carbon cycling, the total amount of carbon that's cycling in the system, and it would seem like some of that, more of that, we're going to end up with some more sequestered as well. Is that a fair way to look at it, or are there too many nuances in there to really kind of go in that direction?
Well, when you build up organic matter in the soil, you're sequestering nitrogen. That is the way to sequester nitrogen. Now, excuse me, sequester carbon, CO2. Now, there are people who want to take ground up rock. Okay, if you take basaltic rocks, basic rocks that are basic in reaction, and you put them in soil, they will convert some CO2 to carbonate. And then the question is, what happens to that carbonate? My guess is that in acid soils, there will be a slight liming effect of doing that, but that you won't really sequester CO2 as carbonate. So my guess is that this isn't really this really isn't something we should be wasting our time with. We're not going to be grinding up, half of the soil's, rock that's available and shipping it all over the world to put in soils to sequester carbon. And in a lot of soils, I'm pretty convinced it won't even do that. It won't sequester the carbon. You can put it in for sure. So I think if you're talking about carbon sequestration in soils, you're really talking about increasing organic matter content, which in some soils is relatively easy to do. I'll talk about that a little bit later. In other soils, it's very hard. In sandy soils, it's hard to increase organic matter content.
Could I just ask quickly a follow-up about mineral-associated organic matter versus particulate, and the capacity of mineral maybe to be a source or a place of sequestered carbon, a more stable form of organic matter?
Well, it is, but it's also stable inside aggregates. So that if you develop aggregates and you don't keep plowing every year and rototilling or plowing and harrowing the soil, those aggregates will remain stable. And organic matter, by the way, helps them stay stable. I'll show you some evidence of that. So that
Thank you.
You want that water stability, the aggregates to remain stable. And if they're staying stable, that organic matter is inside it and it's sequestering it, and that's fine.
Good, thanks.
Okay.
Wonderful. Thanks, Fred. I think those are all the questions we've got on your first section of this presentation. If you want to head on to the next one.
Okay, let me see. Hold on one second. I've got to get back to this. And meeting view, meeting, start share. And do that again. This is what we're sharing. And okay, is that working?
That is, and then if you want to
Yeah,
put
yeah.
it back into the PowerPoint mode, but it does look
Okay.
perfect.
Okay, good. So we're going to talk about, we were just talking about it, but we'll talk about soil aggregation and structure. So, this is a good part of the story of the physical characteristics of the soil. Also, the amount of sand, silt, and clay are important as well, obviously, and they affect aggregation and structure. I'm going to talk mainly about aggregation near the surface of the soil, but the soil structure to depth is also very important. So let's continue. This is Ray Weil, who is a professor at the Maryland, at the University of Maryland in College Park, and he's written the textbook really on the soils, the nature and property of soils. And here's Ray is holding two beakers, and you can see on our left, it's his right hand, he's holding a beaker that had an aggregate, and when he stirred it, and you can see the murky water in there. And then to our right is clear water, but there's an aggregate at the bottom, and he had also stirred that one up. So you have two aggregates here. One of them is very stable in water. One of them will break apart with just gentle moving of the water around. These are some other photos of Ray's, and that's showing, really the two differences in the beakers here. And on the left, on the upper left, you've got a well-formed aggregate holding together in water, and another one just to the right of it. I'm pointing with my finger, which of course you can't see. Just to the right of it is an aggregate which is starting to melt away in water. It just sort of dissolves. It's not really dissolving, but the structure is dissolving. And you can see down below, if you mix them up with a rod or something, low organic matter soil versus a higher organic matter soil in water, there's a significant difference between them. Well, this is what happens if you lose aggregation on the surface, and I'm going to show you some photographs of this in a second. This is from the book. If you have well aggregated soil, and this is mainly surface soil that we're talking about right here, water can infiltrate fairly easily in between the aggregates. It can wet up the aggregates as well. It can get into the soil. And this is what we want. We want water to get into the soil. We want to not have it run off, because if it runs off, it's also going to start taking soil with it. In other words, runoff of water means erosion. You're going to be losing particulates, losing soil with it. And but soil aggregates that are not stable in water will break down and form this surface, which is referred to as a crust. Even if it doesn't seem what you think about as a crust, it can be pretty tough. Water can't enter. I'll show you some photographs of that. This is from Washington State, a wheat field taken in the wintertime. And if you look at that reasonably carefully, you will not be able to see any, perhaps one or two holes. It's like icing on a cake, it's smooth. This is a crust. Water can't infiltrate. When it rains, that is running off. This is why you got this, little rill forming, or this type of erosion going on in the field. So this is a soil that's high in loessl material, and those soils are loess soils without much organic matter can be very susceptible to runoff and erosion. The Yellow River in China, by the way, is yellow because of the loess soils in north central China they are yellow. And the Yellow River is called the Yellow River in Chinese. It's the highest sediment loading river in the world. And it's got all that loading because it crusts like this, and the surface seals, water runs off it, it takes the sediments with it, and you can have a severe erosion problem. Now, this is a soil not quite so bad. This is in Fairfax, which was in corn for many years. Fairfax, Vermont, that is, was in corn for many years. It is now in hay, thank God. But it just shouldn't have been in corn for so many years. I won't tell you exactly where in Fairfax this is. But there was a significant amount of erosion occurring and runoff of that soil. It wasn't a terrible crust, but there was a crust formed on the soil. You don't see many holes. You don't see wormholes or places, old root channels or things like that for roots. Old root channels. This, on the other hand, this is in the intervale, and it's sort of a semi-crust. This the intervale here in Burlington. There are some holes there. You can see some places where it's been sealed by some of the silts in there, so it's not terribly bad. Water will infiltrate, but it's not the same thing as if it was totally well aggregated. So this is not terrible, but it's you'd like to build up some organic matter in that soil. This was us from an experiment that we ran, a runoff from a bare soil on the upper left, lower right runoff from a soil with vegetative cover. And you can see the difference between the two, the more sediments and the runoff from the bare soil, and less so from the one that had vegetative cover. So it's the aggregate stability that's formed by having the vegetative cover there. Okay, that helps to maintain the aggregate stability. This is an experiment that we did, I won't tell you how many years ago, but it was, we added either compost or straw, or grass or manure to a soil, which was this is an average for 3 different soils, sand, loam, and a clay soil. And these were the percentage of water stable aggregates as a percent of the total of this size fraction that we had. So you can see that with manure, we had many more, 60% of the soil aggregates that we had were stable with sloshing up and down in water. In a check, it was like 45%. Compost didn't do much, and we can discuss that if you're interested. This was just a one short experiment, but I don't think compost, it does a lot of good for soils. We use compost a lot in our gardens, but it's probably not the best aggregating agent compared to grass or manure added directly to soil and allowing it to decompose in the soil itself. Again, we come back to this, the soil on the right was these were both from an experiment and where the soil started out the same. Soil on the right had been treated with manure. It was in a good rotation with a hay crop, a couple of years of hay. The soil on your left was in continuous cropping of cash grains, probably mostly corn and soybeans, and the structure of the two was dramatically different. And you could tell, by the way, we had I didn't have a penetrometer or something you can stick into the ground and can tell you how much pressure you have to exert to get into the soil. I'm sure you've seen these or heard of them, but I took a wire hanger and you push it into the soil, and you can tell the difference between the soil on your right, which is much easy to push it in, went in very easy, and it was very difficult to put that straightened out hanger, that rod into that soil on the left. I think this is the last slide for this. One of the things to keep in mind when you're thinking about managing soils is that whatever you're going to use, whatever idea, whatever practice, it will have multiple effects. It doesn't usually just affect one thing. Cover crops, which I'm sure most of you have heard a lot about, and I hope you're going to hear a lot more about it in this course, And there's a whole chapter about it in the book, and there are books written about it also. Cover crops are very powerful, if you will, practice because they have so many effects. And they add organic matter to a soil, you're growing biomass. Either the roots stay there or you're incorporating it in the soil. Enhance mycorrhizal numbers. What happens if the soil is bare during the winter and you don't have anything growing there, your population of viable spores will decline over time. Having a crop there like grass or a legume will allow the mycorrhizae to infect it and produce more spores. If you're growing a legume, it'll add nitrogen. If you're growing a grass, that'll help preserve nitrogen by taking up nitrogen that might be leached out. Suppress weeds, suppress nematodes. If you're using the right cover crops, there's some cover crops that might enhance nematodes. You have to be careful and know what kind of nematode problems you might have. Reduce erosion for sure, increase water infiltration, decrease nutrient loss, and attract beneficial insects. So when you add all those together, this is quite an unusual practice, if you will, in the sense of having so many effects. And that's why so many farmers are turning to use of cover crops, and we should encourage them to do so and to experiment with different cover crops. There's now a huge range that farmers can use of different crops for different situations. If your soil is compact, there are cover crops that might be recommended for that situation. If you do need to produce nitrogen, to add nitrogen for the next crop, obviously you should be growing a legume. Do you use a hairy vetch or something else? There are a lot of choices. Okay, so I'm gonna stop the share, and that should do it. Did I do it?
You did it.
By George, I'm such a technological wizard. Okay.
Wonderful. All right, we've got another question in the chat here. I understand that compost alone will not aid dramatically in building soil aggregates. What about compost in addition to plants, such as cover crops, and especially multi-species crops, in annual cropping systems in the compost, is the compost an aid in this capacity? My understanding is that well-made compost is adding the biological diversity of microbes that is often missing.
Yeah, I can't praise well composted material enough. It's very good stuff. And we use it, like I said, we get manure from a dairy farm near us and mix it with garden waste, and that's really good material. I grew tomatoes just in straight compost alone last summer, just in bales. I got some bales of straw and put them around and filled it up with compost. And those were incredible tomatoes, incredible crop of tomatoes. So that's just straight compost. Adding it to a soil is wonderful. It adds cation exchange capacity, which we'll talk about. It adds nutrients. It adds more biological diversity to soils. So it does a lot of things. And some of them compost, if they're well composted, but not too cured. In other words, not too long after the composting process, can be suppressive of a number of disease organisms. There have been many studies on the issue of composting. The problem with compost and making recommendations is that you can compost anything, basically. I mean, anything that's organic. And so there's no pure, recipe for compost. And this compost is different from that, depending on what you put in it, how many times you turned it, maybe you didn't turn it, and then how long you waited till after it was fully composted. That is, it didn't reheat when you turned it, or didn't reheat very much, how long you waited to actually apply it. So all those are variables which make the whole issue of making a recommendation, just a blanket recommendation for compost impossible. But yes, I would say it's good for you. It's good for the soil. That it's a good way to recycle organic waste. We should be recycling kitchen waste through compost and garden waste through compost, farm waste through compost. There's a guy down in Virginia. I don't know if people have heard of Joel Salatin. Is Joel still active? I once went to a
He
talk.
is.
Is he still active? I went to a talk
Yes.
of his, and he started out this down in Virginia, in Charlottesville, and he got up and he said, "Okay, you folks, you better put your ears on because I'm going to talk fast." And boy, he was off to the races. Well, one of the things that Joel does, he's got some compost, he's got manure, and he's got some other stuff, and he's got it in a barn, and he takes grain and he puts it, like he takes a handful of it and puts it in here, puts it in there, puts it in over there. Then he lets his pigs in. Pigs come in and they are his compost turner. They're going after the grain, and so they're rooting into to this manure and other material to get his compost. Anyhow, I mean to get the grain, and in the process, they're his compost turning system. Okay, a little different than other people. Well, that's true. I use a front end loader on a tractor. We don't have the pigs to do it for us. So, all right. So was there a question in there?
I think you've gotten to the question there. The next question relatedly, and Ellen, I'm not sure if I'm going to capture this perfectly, but Ellen wonders about the making of compost itself as a carbon losing process. If you could speak to that.
Well, you are losing carbon in the process for sure. The composting process is really an accelerated aerobic decomposition of organic materials. That means CO2 is being evolved. If it's being decomposed, just as we're evolving CO2 all the time as we breathe, and of course the organisms are doing the same thing. They're taking in oxygen and they're giving off CO2. The aerobic organisms, as in the soil. The soil breathes just as we do. Carbon dioxide is going into the soil. Okay, excuse me. Oxygen is going into the soil. Carbon dioxide is going out as soil organisms use the organic materials in the soil to live. So yes, there's a fair amount. I don't know what the percentage is offhand, but maybe 40% of the organic material you have can be lost through 30 to 40% just through that accelerated decomposition. So I once had a student who was interested in this issue, and we did some experiment, which I don't have the results here today, but I can describe them to you. We made compost. I think this was using sewage sludge and sawdust, I believe. And we made compost, and we either added the sewage sludge and sawdust directly to soil, or we added compost to soil at the rate of the original that we were at. In other words, if we had, let's say 100, I don't know, let's talk about 100 pounds of sewage sludge and sawdust we add to a soil here, we would add like 60 pounds or 50 pounds of the compost. Okay, it was representative of the original 100 pounds, but since 40 pounds got lost as CO2 went off into the atmosphere, you have to add less material. Everybody with me on that? Okay, so that adding sort of equal original quantity to a soil. And the soils did better from a point of view of cation exchange capacity, from the point of view of structure, from just adding the material just directly to the soil. So should you compost it? I don't know. It depends on the situation you're in. There's nothing wrong with taking organic materials and adding it to a soil. Now, one of the things that composting can do for you, if it's well composted, is it can kill a lot of weed seeds. Now, that's something, so that's an issue with manure if you get it from somewhere. If you're getting manure from a dairy farm, for example, there'll be a lot of weed seeds in it, and we have to deal with that. But composting takes care of a number of them, but I don't know if I do it well enough to get rid of a very high percentage of it. So I think there's good reasons to take material whatever it is, and just add it to the soil and let it do its thing on the soil, either on the surface or mixing it into the soil, depending on your tillage system or whether you're using tillage or not. Can be injected into the soil. There are farmers now injecting manure, liquid manure into soil, which is certainly better than plowing up the soil, putting it on the surface, and then plowing it in. So this is there are different ways to do it. There are different reasons for doing it in different situations. That is, using the materials directly or composting them first and then putting them on the soil. You know, in our situation, I mean, in our gardens, when I'm talking at garden scale now, you know, we're not tilling anymore. And so we're just covering the soil with organic materials, leaves, grass clippings, stuff like that, put compost on. So we can use materials directly. That's another way to use it, as you know, as a mulch. I forgot to put on the cover crops. Another thing you can do with a cover crop, if you grow cover crops in between your rows of crops, you can what they call mow and blow. You mow the crop and you blow it onto the next row, and you can create a mulch that way. So that's another thing you can do with a cover crop, is actually mulch the next row, shall we say, over, which didn't have a cover crop in it. Again, some digression.
No, this is great, Fred. Thank you. We've got a question from Tom. Many pasture managers promote the concept of livestock trampling of partially or ungrazed forage onto the soil surface or perhaps into the soil as a means of increasing soil organic matter. Do you have any sense of how much of this organic matter is respired off from the soil surface versus incorporated into the soil?
I really don't. I mean, it's a good question, and it's. I mean, all the questions have been good, by the way. It's a good question. Certainly, intensive rotation grazing or management intensive grazing, MIG, goes by different names, is a very good management tool for getting the maximum amount of pastures, and it's healthy for the animals. They're always eating really high quality forage, and you're getting them off quickly, so that you give the plants time to regrow, and so it's a very good system. I'm convinced. Now, how it affects the soil? I mean, in general, under pastures, you should be getting an increase in soil organic matter, and it may be slowly over time, but it's happening. Now that the pushing into the soil, you could say, well, you know, if it pushes too far into the soil, it may be causing compaction. So if you got, you know, cows on a field when it's very wet and that soil is prone to compaction, that may not be good at all. You know, you may be causing, you know, the pock marks that you see in soils. I made a big mistake last year. I'm confessing to one of my sins. Actually, this was a year and a half ago. My blueberries this last summer was incredible blueberry year in Fletcher. Okay, I won't say it was anywhere else, but I got more blueberries than I've ever had before. The year before was terrible, and I think it had to do with the you know, the coincidence, or the poor coincidence, if you will, of pollination with flowering, that the insects weren't out, the bees and the others that do the pollinating, because of cold weather. But we had a frost in the middle of this flowering this summer, and it was just incredible. Well, the year before, I decided I'm going to get back, and I don't take care of my blueberries the way you should. So what I was going to do is let my cows in, a neighbor's cows that graze on our place in the summer. At the end of the season, I was going to let them in and let them just, you know, go through the blueberries and clean them out. You know, just clean out all the crap around them, which they did. I mean, it was not a terrible idea, but they left a lot of pock marks in there because it was wet, and so I think they did some good, but they did some bad too. It made it more uncomfortable for me to walk over and to try to harvest the blueberries, let alone to try to mow the grass in between the rows. So that's anyway something to keep in mind, that you want to keep cows off when the soil is wet, just like you want to keep tractors off the soil when the soil is wet. Martha Stewart even says you shouldn't walk on your garden when it's wet. And you know, Martha Stewart knows everything about everything. And she's right. You know, don't get on your soil when it's too wet. So I don't know. It's a long-winded way of saying I don't know the answer to that. My guess is it's not a huge influence, but it's part of the process, and the process itself is good for soils.
Thanks, Fred. And then we have someone on the chat, Ellen asks, any thoughts about using wool pellets as a soil amendment?
I know they're used, but I have no thoughts about it whatsoever. I don't know. I have no idea. I won't even guess.
Okay.
I don't know.
Um,
I'll get into more trouble than I'm already in.
and then I guess I have a question for you. I guess maybe two questions. One is just you mentioned adding a leguminous cover crop is important for the next crop. And I think that that's a really important distinction that sometimes gets lost, that if you plant a legume, you're not adding nitrogen to the crop you're growing, but it's really available later. Is that correct? Once that plant dies?
Right, right. I mean, there is some nitrogen that will go from a legume to a non-legume, which is right next to it. Okay, so there is some transference of nitrogen, maybe through mycorrhizal hyphae, or it may just go through the soil, or roots. During the growing season, roots die and new roots grow, and there are microorganisms that are working on the roots that have died. So that but there is proof that there's transference, so it can be transferred. But generally, you don't get a lot of transference that way. I ran into another digression. I was in South Africa at a farmer's field. These were, I think it was all women farmers in this particular place. They had plots like a hectare, like a couple acres, a little over a couple acres. And I got out with my notebook, and here's this woman. She got out her notebook. She had her notebook out. She had a pencil there. She wanted to know my name. I want to know her name. Wrote it down. And they were doing experiments. And one of the experiments they did was they were growing corn, and she grew a legume alongside, right next to the corn. And I've forgotten what the legume was, some clover, I expect. And she wanted to know whether it was going to help the corn or not. And I asked her, what did you think so far? She thought so far it wasn't making any difference at all. So that was her belief. But yes, it's for the next crop. But the next crop can be immediate. You can I've got videos of people traveling through mowed down vetch or crimped vetch, and they're planting corn behind it. So it's the next crop, but it's immediate. Okay, you're planting it immediately. Of course, not all of the nitrogen becomes available immediately in a no-till system like that, but it can happen fairly quickly. So that's another thing to keep in mind with cover crops. If you're going to let the cover crop grow only in the spring, there are a lot of farmers, dairy farmers, that are planting winter rye, I guess it is, and cereal rye, and it gets I don't know, 7, 8, 9, 10 inches tall, and then they harrow it in or plow it in or whatever, and there's hardly any biomass in that. So don't think you're adding a lot of organic matter. It's better than not doing it, but because you're holding the soil intact, it helps with regard to infiltration. You've taken up a little bit of nitrogen that may be recycled, but you're not adding a lot of biomass to the soil. You've got to have a really good, few feet of a crop to be adding significant amounts of biomass. So it's another thing to keep in mind with regard to cover crops is you got to let them grow if you want the biomass. Now, some of them, of course, winter kill. It's another story. Yeah.
Actually, sticking on that theme, I'm wondering if you could speak to, I believe in your book, you speak a little bit to this, having the cover crop, even if you kill it with pesticides, is still better than not, because you have that active soil life to kind of process some of the pesticides. Is that correct?
Yeah. I mean, I think cover crops is a good practice to use for all farmers. It may not fit for everybody, but it can fit for most. I don't see why it can't. Uh, and uh, and there are ways to kill them without using pesticides. You can do it if you mow it at the right time. It may work. You can suppress it enough with this rolling crimper that they developed at the Rodale Institute, which you can find online easy enough, and you can see it in action, and that works quite well too. There are also cover crops that winter kill. I mentioned before, oats will winter kill. The tillage radish, if you have a compaction problem, then you can get a tillage radish on in the fall. It'll grow a nice root down, and it winter kills. It's very good also for dealing with weed problems. So it really helps with weed control, even though you're not growing a lot of biomass in the spring. So there are ways of dealing with cover crops without using pesticides. Well, herbicide in this case. But so, but if you're going to use, if you are in a conventional system and you are using pesticides, and you ask me, should I be using planted cover crops? I would say yes. Plant them. Sure.
Wonderful. And one more question from me, and then if anybody else does have more questions at this point, please toss them in the chat. Concerning the graph that you had earlier, where the manure, the compost, and the straw, and all the different items, I guess I work with a lot of farmers who use liquid manure or a bedded pack system or a composted bedded pack system. And I guess I'm just wondering if you have any thoughts on the different types of manure and how they're applied, and how it could lead to compaction, or if there's anything better than another, or if there's any issues with using a bedded pack system that has so much, in theory, lignin or cellulose.
Yeah, I haven't worked with bedded packs. They look awfully good. They look, just from the farmer's point of view, easy to manage in the winter because you're not doing anything. I mean, you don't have to deal with the manure. It's just the cows are getting taller. That's all. Through the winter, they're getting closer to the ceiling as the pack accumulates, and then you got to go in and clean it out. Yes, it is a different form of manure. The old style manure, like from a stanchion barn, had bedding in it. It had hay in it also. I would say it's something similar to old type manure, except maybe more carbonaceous material inside. For the amount of, let's say, one cow's worth of manure for 1 month of bedded pack versus one cow's worth of manure for 1 month as liquid manure, the earlier, that is, the bedded pack one, is going to have more potential organic matter effects on the soil because you're adding more organic matter, you're adding more materials in addition to the manure per se. In the liquid manure, there's a lot of farmers aren't using bedding, or if they are using bedding, it's there's a farmer near us using sand, and they recover the sand, and so they try to recover most of the sand, and the liquid is pretty liquidy. So you are adding carbon to the soil for sure, but not as much as if you had been using a bedded situation for cows. So they are different. One will have more nitrogen available. The liquid will have more nitrogen available right away, but it should be plenty of nitrogen in there, but it might be not quite as available as rapidly. But certainly bedded pack manure should be quite good for providing nitrogen. I wouldn't worry about, unless there's an incredible amount of carbon as residue, that is, as straw in there. I wouldn't be too worried about it. You can overdo it, of course, and immobilize the nitrogen that's in the manure. Anyway, sort of beating around the bush.
That works. We did get another question from Julia, and she's asking, what cover crops do you usually recommend to farmers in Vermont, if any?
Well, it depends, what crops you're growing and what window you have of available time. What are your objectives? What are the issues that are on your mind, and what's the time period you have available. And I think, the rye cover crop is very easy to use. It's very easy to get it established. It will if you want it to overwinter, it'll overwinter, and you can grow some pretty good biomass in the spring. So, I mean, that's not a bad gracious if you just want grass. On the other hand, there are people that are having tremendous, and of course, you can plant legumes. I mentioned vetch, but there are other legumes you can grow. And there's some people that have had very good success with mixtures, and they just try different ones. And I think it's important for farmers to experiment on their own soils with different practices, not just talking about soil. Whatever they're doing, they should be experimenting every year, trying something a little different. And if you think it's good, try it on a little bit larger area. Don't take your whole farm and do something to it and then say, "Oh my God, I shouldn't have done that." So I mean, I would try some of the mixtures and see what works. But the key thing is, what's your window? How much time do you have between terminating your last crop, that is harvesting the last crop, and planting the next commercial crop that you're going to grow for production for sale. So if that's a very short window, you don't have too many options. You probably want a grass. If it's got a longer window, you got all sorts of options in there. Again, it depends. Like I said before, do you have a compaction problem? Are you trying to add a lot of nitrogen? Are you trying to just add a lot of organic matter? All these questions. That's discussed, by the way, in the cover crop chapter in the book. And there are other discussions of cover crops, in there, in other places.
Wonderful. Thank you very much. With that, if you want to move on to your third section, or
Okay.
maybe get a glass of water, up to you. I know we've been peppering you.
I was thinking of letting my dogs out there. They're in the back room here, so they shouldn't. You've heard them probably barking a little bit, but not much.
Not much, no.
Now we can start.
Okay.
Let me get out of this. Get back to that. Get to here. Start share. Okay, and then I've got to go to here.
Looking good.
Okay. Everybody got their ears on? Okay, cation exchange capacity. Well, I presume this doesn't come as any great shock to you, but soil particles have negative charge. There's a lot of, when you're walking on a soil, it's not going to affect you. It's not totally, the soil itself is neutral, but the particles of clay and organic matter have negative charge, which enables them to hold on to positively charged ions, which would be cations. And we'll talk so what I'm going to talk a little bit about is, where does this come from and how is it related to soil pH? And a little bit, maybe, I don't know if I'm going to talk about how it's measured and how can you increase it. And cation exchange capacity is considered to be a very positive thing. You want to have a good store of nutrients. And it is a storehouse of potassium and calcium and magnesium especially, and hold on to ammonium, but most of the nitrogen that's available to plants in our soils is there as nitrate, which is negative itself. So it's not held on to these particles. This is something I think directly from the book. It can be held on humic materials, that is on soil organic matter. These are, whoops, I'm pointing with my finger, of course. Here's a calcium ion, a potassium ion, and a calcium ion over there. It can be, these cations can also be held on the surfaces of clays. And the example soil that I was sent by Lauren, it definitely has some significant amount of clay in it, and I'll explain how I know. And then you have also organic molecules, which are chelates. That is, they can hold on to, this is in this case, it's zinc, which is a micronutrient needed by plants. It's an essential nutrient, but needed in small amounts. We need it too, by the way. Animals need zinc. And if it's held by more than one bond by an organic molecule like this, it's considered to be a chelate, and you can buy organic chelates. They use them in the greenhouse trade. You know, if you're making a hydroponic solution, you probably want to chelate your micronutrients, or at least some of the micronutrients. So the significance of cation exchange capacity, or CEC, is that it holds on to nutrients that we want held in the soil for plants to use. Okay, they're held relatively lightly on the plant, and they can come off. In other words, that this calcium can come off, and another potassium can go on there, or something else, so that this is exchangeable. In other words, it's not completely fixed surface. It is exchangeable. Come on, can go off under the right conditions. Okay, there's a lot of weak acids in soils, and I'm going to talk about what I mean by that. Mostly they're associated with soil organic matter. So I'm going to get into a little bit of chemistry here that if it bothers you, you don't really need to worry too much about it. I'm just trying to explain where it comes from. So that basically, if you add a limestone to a soil, which is a base, you have an acid soil, means a low pH. You're adding limestone to the soil, and you're increasing the pH. In that process, you are pulling hydrogens off of the certain organic sites that are weak acids. There's also carbonic acid in soils, which is H2CO3. You can produce it if you got a straw and you blow into a glass of water. You are producing carbonic acid because the CO2 in your breath combines with the water and produces H2CO3. And some of that hydrogen, if you add a base to this, some of that hydrogen will come off, and you'll have HCO3 negative plus a hydrogen, which then gets neutralized by the base. So that's a weak acid also. That's a weak inorganic acid. But in soils, what we have is a lot of organic acids. But they're weak in the sense that that hydrogen isn't readily coming off. It's not, like a strong acid, well, HCl, hydrochloric acid, it's dissociated. The hydrogen is separated from the chloride, and boy, you stick your finger in that, and it'll burn it off. Carbonic acid is a very weak acid, doesn't produce a lot of hydrogen. So a lot of the hydrogen stays connected to the original molecule. Okay, so organic molecules are just carbon chains that have different groups attached to them. In this case, these are hydrogens, and then they are at the end. This is the organic acid part, a double bond to an oxygen over here, and a single bond to an OH group, the hydroxyl group. And that hydrogen from that hydroxyl group can come off if you add base. If you add something to provide an attraction to it, if you will. It's not exactly that, but close enough. And then potassium or calcium or magnesium can take its place, so that this is really never completely negative. Okay, it's maybe negative for a very short period of time. And then you have this hydrogen, which is neutralized by the base that you're adding, and you get water. Okay. If you're adding hydroxyl, or if it's H2CO3. Anyway, I won't get into too much of the chemistry. But the point is that organic matter has a lot of these types of organic acid molecules, basically. And so there is a lot of cation exchange capacity associated with organic matter. I'm trying to give you, all right. I didn't realize I'd forgotten that I did that. Before I get to that, what I said I was going to tell you, it's important to keep in mind that clays also have cation exchange capacity. And some clays have as much as cation exchange capacity is expressed in the actual number of charges per 100 grams of soil. So this is milliequivalence, which is a number of charges. So that's You know, it's like weight would be milligram, would be a thousandth of a gram. This is a thousandth of an equivalent, which is 6 times 10 to the 23rd molecules of a charge, positive charge. Okay, so, or in this case, negative charge. So some clays, in other words, if you had 100% clay, there would be 100 milliequivalents of charge in that soil. If it was 50% clay, there'd be 50 milliequivalents of charge due to the presence of clay. Okay, everybody with me? Okay, there are other soils, there are other clays that are very low. Kaolinite, which you find down in the southeast United States, has a very low cation exchange capacity. So you can have a clay soil and it's still not that high amount of organic matter. So it's not a half of this. It's a 10. Well, it's 0.05 instead of 1. So that if you had 100% clay, you'd have 5 milliequivalents of charge in this soil for 100 grams of soil, because it's 100 grams of clay. So just move that over to 2 places. Again, I'm pointing with my fingers, but you can't see. Okay. Clays in the Champlain Valley, the types of clays that we have that are mostly lake deposited tend to have around 0.2 milliequivalent for every percent clay. And so that's a reasonably good number if you want to play around with these numbers and have some estimates. So if you have a 50% clay in your soil, 50% of 50 grams of clay for every 100 grams of soil would be 50%. Okay, 50% times 0.2 is what? No, 50% times 0.2. If it was 0.1, it would be 5, right? And if it was 0.2, it would be 10. So you'd have 10 milliequivalents of charge because of the clay in that soil. All right. So that's a milliequivalent for every percent clay. Now, something also that complicates things is clays with soils that have higher clay content tend to have higher organic matter content, because the clays provide, the organic matter can adhere to clay particles, that is organic molecules, and also they have greater amounts of structure, that is more aggregation in clays, and you sequester more carbon inside these aggregates, and that's why clay soils tend to have higher amounts of organic matter. So you go to what used to be called heavier, which is the term maybe farmers still use, from sands to loams to silty loam to silty clay to clay, you get more and more organic matter in those soils naturally. Okay, and we can talk about it later if there are questions about why. Now, for many soils, organic matter provides a very large percent of the CEC. Okay, around 80 to 100% in many soils. May only be 50% in some soils, but it's still a high percentage of the total ability to hold on to calcium, magnesium, potassium, ammonium is because of the organic matter in the soil. And organic matter provides a large portion of cation exchange capacity, even for some fine textured soils, for heavy soils, so-called, the old way of talking about it, that contain clays that don't have a lot of cation exchange capacity. So that a high percentage of what's there is due to the organic matter, even though you've got a lot of clay in it. So, and I'll give you my guess about this sample that I was sent when we talk about soil tests. So let's see. So how do you increase cation exchange capacity? Well, basically, there's only 2 ways. You increase the soil organic matter in your soil, which increases these organic acids, which are the site of so much cation exchange capacity, or you increase the pH. And as you're increasing the pH with lime, usually lime ash mix, if you want, or ash by itself, wood ash, whatever, if you increase the pH, you are pulling off hydrogens from organic acid sites and allowing them to hold on to calcium or magnesium or potassium. Okay? So there's no other way really to increase cation exchange capacity in the soil. This is it. If you find another way, let me know. Okay. Okay, this is a chart. Well, let's put them all there. Let's get it all out there. Okay, this is on the left side. What you've got is the cation exchange capacity due to organic matter. This is per 100 grams of soil. Okay, and down below is the pH going across the bottom in the horizontal axis. So let's look at 5% in here. Just why not? If you start at a very low pH, like 4.5, which is very low for agricultural soils, you start with a relatively low cation exchange capacity. But look what happens if you get to 6.5 pH, 6.5. You've got about 10 milliequivalents of charge due to the presence of organic matter in that soil. Okay, forgetting the clay content. The clays may add to that. Okay, but this is just due to the presence of organic matter. So we may come back to this, or I don't know how clear this is. I believe this is in building soils. I'm not positive. I believe it is. This charts. Some of these I've used in different places, but you can see it at pH 6. Just says, look, if you have, well, let's take 6.5 where we were. If you got 7% organic matter, you've got like 14 milliequivalents of charge due to the presence of organic matter. I don't know what happened to my, oh, here it is. If you're at here, you've got about 10. If you're at 3%, you've got about 5. If you're at 1%, maybe you've got, 2 milliequivalents of charge if you're lucky. All right. Keep in mind, and we're going to have questions just in a minute or two. So if there's anything here, and we can go back to this, but all this is showing you is that different amounts of organic matter in the soil can give you vastly different cation exchange capacities, and these change with pH, dramatically with pH. So that liming an acid soil creates cation exchange capacity. Adding organic matter to a soil, which is going from here to here to here to here, increases cation exchange capacity. So this chart should just be showing you what I told you just before. All right. Let me see. Okay, well, anything I want to say here before the Q&A? Not really. Let's see what your questions are, and then we can go back if we need to. Let me stop share. Okay, we okay?
We're doing great. You look good.
Okay, that's a relative term, of course. Okay, so people have questions about that. I know there's a lot of stuff coming at you fairly quickly, but let's go ahead and see.
So I'm not seeing too many questions in the chat. I guess one question that I would have is the calcium and all of the positive charges in the soil. Where are those coming from to react with the cations?
Basically, it's coming from, well, originally it came from the decomposition of minerals in the soil. The minerals that are in the soil are dissolving in an acid situation. The soils in Vermont, by the way, are naturally acid. We have very few. We have a couple of soils that I've run across in the Champlain Valley that have limestone in them and are not naturally acid. But most of them are naturally acid, and if you get up into the mountains, quite so. So that so in acidic situations, especially, minerals dissolve in water, and especially in acid water, they will dissolve, and that's where you're getting your nutrients in your soil originally. Now they're being cycled also. There's nutrient cycles. We take nutrients out of the soil into the plants. We put residues back. We put manures back, and we're raising animals back into the soil. So we're cycling some nutrients we're getting in that way. A lot of them just from the minerals in the soil. And of course, then people are buying fertilizers and putting them on soils. And so you can get them that way, or can import compost onto the farm or import manure onto the farm. I mean, like we're importing some manure onto our gardens. It's not, I wouldn't call it a farm, but we're importing it from off the farm. So that's the way. It's either cycling, it's coming from the minerals originally, cycling, or you're adding it from somewhere else, from some other source, fertilizers or organic materials that you're getting from somewhere else. So, but that's basically it. We have a lot of phosphorus in our soils. By the way, our soils tend to be not that bad in phosphorus. A lot of the soils, especially the sandier ones, have mineralogy that is such that they have good amount of phosphorus in them. It's just the luck of the draw. It's just that they happen to have high phosphorus minerals in them. The soils up, I'll show you in a second, in a bit when we get to the soil test. The soils up in Franklin County tend to have much higher magnesium contents relative to calcium than to the soils in Addison County. So I assume that's a difference in mineralogy, higher in minerals that are high in magnesium, like chloritic minerals are higher in magnesium than the minerals that you might find as average in Addison County. Now, why? I presume it came out of a different watershed, coming out of the mountains way back when from the Lamoille River versus the Winooski and from further down into. But that's where they come from, basically. And there's new minerals being released all the time, and organic acids are being produced by microorganisms in soils, and they are helping to solubilize minerals like phosphorus minerals. A good part of that becomes soluble during the season because of the activity of life in the soil. So there are different ways that it's coming, but it's coming originally from the minerals. And if it's not coming from the minerals, you're importing it somehow, or it's coming from the sky. Okay, we used to get a lot of, well, it's not a cation. We used to get a lot of sulfur here in the Northeast because of the air pollution from the electric generating plants out in the Midwest. Well, now they've cleaned up the plants. Our air is cleaner. There's less acid rain, but we're getting less sulfur coming in the rainfall. So you'll see that the soil that you sent me, the example, it's on the low side with regard to sulfur, which we probably wouldn't have found 20 years ago or 30 years ago. So, and of course, anyway, so that's the cations are coming from the decomposition of minerals.
Wonderful. We got a question from Charles. In low clay percentage soils, high sand percentage, does the relationship change between pH, soil organic matter levels, and CEC?
No, it doesn't. That last graph that I showed you, which showed the 1% slope, that is basically a sandy soil. Sandy soils tend to have relatively low organic matter. It's very hard to increase the organic matter, especially if you're tilling the soil, okay? Because you're exposing it to the organic matter that's there to decomposition more readily, to more readily decompose. So it's not, the relationship is there. The thing is that it's much more difficult to increase the organic matter in a sandy soil because it's so porous, it's so oxygenated, and it doesn't have a lot of particles to help stick together and make aggregates. Do you have aggregates in sandy soils? Sure, you can have aggregates. They're probably not great aggregates. They're not very large aggregates usually, because you don't have the clays and the silts to help stick it all together to make it cohere as an aggregate. You can take a lot of sand particles and put them together. You say, well, I've got an aggregate, but let go of that and put a little water on it and poof, it'll melt away. The aggregate's gone. The sand particles are just individual particles. So that it is difficult to increase the organic matter. But it's true, if you're able to increase it to 1.5%, which is doable, adding a lot of organic matter, reducing tillage, yes, it will be adding more CEC to your soil. You have probably increased your CEC by 50%. You're going from 1 to 1.5. It's an increase of 50%.
Wonderful. Um, I know in your book, and you slightly mentioned it earlier, the addition of ash or lime. I know biochar is quite a big topic these days. I didn't know if you want to take a minute to speak about biochar and what you know or what questions might remain there.
Well, biochar is fine. If you want to put biochar on your soil, fine. You run into the same issues that one of the persons asked with regard to composting. As you're producing biochar, there's a lot of CO2 going off into the atmosphere, but you're burning it. You're burning it under low oxygen conditions, so it's not producing complete ash. But I don't. The experimental evidence is. Let me just put it this way: there's contrary experimental evidence that it's inconsistent. Some cases it seems to help, other cases it doesn't seem to make a difference. Some cases it may be harmful. Don't forget, in that biochar, you've also got ash. So I mean, wood ash is good. We know it's good. It has a liming effect on soils. There is potassium. There's calcium in the ash. There are other, nutrients that plants can use that are in the ash. So that ash is good. We know that. We've done experiments. Farmers have used wood ash for years. It's good stuff to use. It's good liming material. And so you've got ash mixed in. So how do you separate the effects? I don't know. So to me, the char it's a side issue that is not terribly interesting. If you want to use it, fine. I mean, if it's your farm, go use it or experiment with it. That's the best thing. If somebody asks about it and they're gung ho, they really are interested in it, and I say, listen, give it a try. But if you really want to know for sure about something, you can't just put something here and not put it there. You got to lay it out like maybe strips. Yes, here, no, here. Yes, here, no, here. So otherwise, you put it there and you become convinced. People become convinced very easily that something they did that they thought was going to help, that it really helps. And this is a major mistake that people make in general, and farmers make it in particular. And that's one of the reasons that farmers have been feeding so much more phosphorus than their cows need, because there was a mythology that cows needed that extra phosphorus. I'm talking about milk cows, dairy cows. And it turned out not to be true at all. And it was recommended by folks to do it, and farmers believed it, and it turned out just not to be true. So we were overloading our soils with phosphorus. So anyway, my point is that if you want to try something, whatever it is, try it on small scale and replicate it. Even if you don't do it as a scientific experiment, put in a few different replications separated by not doing whatever it is that you're trying new, and see what happens. And if it works for you, try it on a larger replicated one next year and see if it happens there. So biochar, I'm not convinced that it's environmentally meaningful. I'm not convinced it's really going to help everyone. It may help some people, but we don't know exactly who, and it may hurt some, and we don't know exactly who there either. So, it's okay. Probably won't do too much harm.
Thanks, Fred. We've got a question from Tom. Could you please compare the liming action of ag lime versus wood ash? Are they equal?
No, because, well, on a volume basis, wood ash is more light. Okay, so there's less per wagon load. There's less neutralizing ability in the wood ash than in the limestone. And you have to account for that if you're going to use I don't know if they're still selling it, the lime ash, the limestone wood ash mix was being sold out of, what is it, Swanco, right? Swanton Lime, I believe. No? You guys
I'm
should,
not sure.
somebody should know. Anyway, but they were being used. They were using the wood ash from the McNeil generating plant here in Burlington, mixing it with limestone and selling it. I put some on my pastures. It seemed pretty good stuff. And that was many years ago. I probably need some more. But it's anyway, so by volume, it's less. by weight, it's probably, I don't know. I don't know for sure. I'd have to go back and check, but we did do research on it. It's good stuff, but it has less neutralizing value by volume, certainly.
Thanks. Willie, do you want to ask your question out loud?
Sure, I'll just mention also, I've got a little background. Wood ash and lime ash. There is a stockpile of lime ash still in Shelburne at SLC. They're not making it anymore, but it can be acquired if somebody wants that particular mix. Wood ash is usually somewhere around 900 pounds per cubic yard. So it's, whereas lime is typically like 2,000 pounds per cubic yard. So that's your density thing that you were talking about, Fred. And then you've also got the amount of calcium usually in a ton of wood ash is around 100 to 150 pounds, 50 to 75 pounds maybe of potassium. So got other minerals and stuff associated with it. Usually it's going to be somewhere around a third of the calcium carbonate equivalent of most of your good limestones. So my question, Fred, I just wondered if with today's topics, which are great, that you've been addressing for us, do you want to talk about them at all in regards to specifically the context of the rhizosphere at all? Is that something you're interested in bringing up at all? It's a different place, different world in a way almost, it seems like. So I just didn't know if you'd want to talk about that.
I'll mention just one thing, and then we should get on because soil biology is going to be dealt with in a whole other session. But everyone's aware now of the human microbiome. We've got all these organisms living inside us and living on our skin and whatever. So, and it's very important for our healthy functioning to have a healthy microbiome. Well, it's the same thing for plants. They have a microbiome just like we do. They have organisms living on their leaf tissue, on the stems. There are organisms living in the plant. There are organisms living on the roots. And that's the rhizosphere that Woody was talking about. And it's terribly important. It's a hotbed of activity of microorganisms, and we want them to be there, and we want them to be productive and healthy. And so that mycorrhizome, for that to be a healthy one, is what we're striving for. If we're talking about soil biological diversity and soil health with regard from the biological point of view, yes, we want a healthy microbiome there. And as by the way, organic matter decreases in the soil, the number and variety of organisms in the soil decreases. And so the soil microbiome that's available to the roots becomes less diverse. You have more problems with nematodes, more problems with diseases, etc. That's part of the reason.
Awesome. I got one more question for you here, and then I know and then I'll have about 20 minutes left for part 4, which
Okay.
you can feel free to speed through or cut out. So the question from Hannah is regarding non-point source phosphorus loss. Where is the sweet spot in the relationship between pH and organic matter content that ensures maximum plant growth and still limits excess P loss?
Well, I don't think that it's an issue of pH mostly. I think the issue, of P loss from farms. I think the greatest issue, I don't know if it's being attended to, is that a lot of the phosphorus loss comes from a relatively small percentage of a farm, usually. And there are certain places on the farm that produce a lot of runoff. And and these are the places on the farm that should be emphasized. I mean, this is something that, should be part of the program. I don't know whether it is. And the way to find out those places is to go out when it's raining and look and see where that water's running. Where is it coming from? Where is it pooling? Where is it starting down the hill? And where is it coming into the stream or the ditch on the side? And those are the places to concentrate on. So if we want to do that, I think that's the biggest issue. The other one is don't overload your soil with phosphorus. And we've found that soils have been overloaded with phosphorus, mostly Some of that's imported feed. These are dairy farms, but you can have organic farms that overloaded their soils with compost and have too much phosphorus in them too. So I mean, that's another option. But certainly, the sediments that contain more phosphorus will be more polluting than the same sediments if they contain less phosphorus. So it's a combination of slowing down the runoff, which will slow down the loss of particle material from the soil, and then making sure that the particle material that's leaving the soil is reasonable with regard to phosphorus concentration, that it's not too high in phosphorus. Those, to me, are the main key factors with regard to phosphorus.
Wonderful. And Hannah, I saw you guys came off mute. I don't know if you want to add anything else to that question, or Kent, looks like.
No, I just wanted to add my thanks, and great to listen to you again today, Fred.
Wonderful. Thanks, guys. All right, Fred,
Okay.
part 4. Keep in mind, we will have another full course on the soil tests if you do want to speed through some things. I apologize.
Good to know.
Yes.
Let me do this, get to that, start sharing. I'm going to become a professional at this.
You are. You're going to get asked a whole lot more.
Okay, then I got to take this and start the slideshow. Okay, where can I do that? No, I got to move this. Here I'm bragging, and now I can't. Oh, here we go. Okay, good.
There you go.
I got George. Okay.
Perfect.
I'm going to skip a lot of slides. Don't panic. Lauren has them if you want to see them. Also, you can send me an email or contact me if you have questions about them. But keep in mind that nutrient testing is really just one part of nutrient management. Okay, so there are many other issues about flows of nutrients. I just talked about a flow of nutrient onto the farm with regard to phosphorus. So much phosphorus coming onto farms as dairy farms as feeds, and importing phosphorus fertilizer as well. You have nutrient cycles that are part of the story. You have organic matter management. Okay. Let me try to take some of the mystery out of soils, but I may soil testing, but it may end up being more confusing. Okay. There's lots of confusion about soil tests. And that's partially because different laboratories use different procedures. And basically, there's an infinite number of liquid solutions that you can shake a soil with. Okay, you put it in a bottle, shake it up, and you can come up with all sorts of acids and bases and organic and inorganic. You can shake a soil up with anything. You can shake it up with water and then filter it and see what's in it. So that different labs use different chemicals. They have different procedures. I'm talking about for extraction of the soil, different ways to report the results, and there are different recommendation systems. So because of this, it's important to stay with a reliable laboratory. Don't keep switching laboratories from one to another, because if you do, it's just going to get you more confused. Unless there's a good reason, unless you feel that they're not doing a good job. There is a lot of confusion about this. I mentioned different solutions being used. This is for phosphorus. These are different solutions which are used in different places to extract for phosphorus. Now we're using a modified Morgan solution, which is this one in the middle here that with a yellow circle on it. These others will work on other soils. Some of them will not work very well for our soils. This is in different regions. This is just showing you that in different regions, they use different ones, different soil tests. There are different ways to report the results, okay? You can report them as pounds per acre of potassium or calcium or magnesium. We're reporting it as parts per million. Pounds per acre would be just twice that, because it's considered to be 2 million pounds in a acre to 6 inches of soil. So that would be index of 1 to 100. Some labs report them as an index. They don't give you form. It may be reported as P or K, or it may be reported as P2O5 and K2O. One thing to keep in mind, if you've heard of or used fertilizers or seen results with P2O5 and K2O, you may have a shock coming to you. There is no P2O5, there is no K2O in our soils. There is no P2O5 and there is no K2O in any of the fertilizers that you will use. Okay, so why do we use those numbers still, or that way of expressing it? Well, if I was a fertilizer salesman, I'd rather be selling you 100 pounds of K2O5 than 80 pounds of K. That's the same thing. K2O, I'm sorry, as 80 pounds of K. I'd rather be selling you 100 pounds of P2O5 than 44 pounds of P. That's the same thing. It's the same amount of phosphorus fertilizer. So the fertilizer industry has a lot of reasons they don't want to change, and farmers are used to it. So anyway, then there are different ways to take the results and then make the recommendations. Hopefully, you've got a good soil test on the basis to make the recommendations on. And what's a good soil test? A good soil test is one in which if it's very low, if it's down here, the soil test is low, you will get a good chance. This is the yield that you get without adding any fertilizer at all versus what you get by adding fertilizer. So you'd get, let's say, 65% of the yield than if you added fertilizer. Whoops, went too fast. Okay. And if your soil test is very high, that means by adding fertilizer, this is called the relative yield. You don't get any increase. You're getting at 100% by not adding anything. Here you're getting 80% if you don't add anything. And here you're getting 65% if you don't add any fertilizer, phosphorus. So this would be a fairly good test. This is a very good test if it was exactly that. The real world is more messy. And this is our data from the PSNT. This is the nitrate test that's done in the summer, separate from the other tests. And this is the way data looks. But this is a very good correlation. But it's the same thing it's showing you. At very low nitrate levels in the PSNT test, you have a relative yield which is low, which means there's a high probability of getting a response by adding nitrogen. As you get higher and higher and higher, you get a low probability. This sometimes shocks extension people and it sometimes shocks farmers. We're dealing with probabilities here, okay? It's not an exact situation. This is just some data from Iowa showing you their phosphorus, two different ways of measuring phosphorus in their phosphorus test, but basically the same thing. There's a lot of spread around. You've got some soils that are test relatively low, and you still get, and they do fine without adding anything. The relative yield without adding anything is fine. Others that are about the same soil test level, but their relative yield indicates that, they need some phosphorus being added to them. So what I'm trying to show you here is the data behind, which means that you've got to look at all of this with a certain amount of respect, shall I say. We're dealing with probabilities. So we're really saying at a low soil test, you have a high probability of getting an increase in yield by adding fertilizer. If it's a high soil test, you have a relatively low probability. We'd like our soil tests to stay around, the medium range, not go high, not go low. If you're over fertilizing, it'll keep going up and up and up. And we've seen this with phosphorus fertilizer for sure. If you're under fertilizing, your soil tests will keep going down. You may be going a little bit over, coming down with a corn hay rotation where you fertilize the corn more heavily than you fertilize the hay, or don't fertilize it at all. I'm going to skip that. I'm going to skip this. I'm going to skip this. And I'm going to go to this. So this is a soil test that Lauren sent me. I've never been to the field. I don't know. Is it a real soil? Is it a real soil test or did somebody make it up?
It's a real soil test. I just took
Okay.
off the identifying features.
Okay, good. Fine, that's fine. Anyway, if we look at it, we can see that the pH is not bad. It's about 6.5, which is the range for most crops that we're growing here in Vermont. You can see, oh, up here it even shows you, it's a little cheating, shows you the phosphorus is on, well, it's a low medium, the potassium is medium, and the magnesium is high. And as soon as I saw this, I saw the magnesium being that high, is I figured this is a soil from Franklin County, which I presume it is. Is that true? It is, yeah.
That's true.
Because in Addison County, you wouldn't normally have those soil tests that high. This is in parts per million. This is how it's being reported now. We used to report it as parts per 2 million. So you take all those numbers and multiply by 2. Well, let me just look at some other things that are interesting here. The soil organic matter is 4.5, is almost 4.5, 4.3%. The cation exchange capacity is 14.5. That would be milliequivalents of charge per 100 grams of soil. And that's not bad. That's a pretty good organic matter content. And I would estimate that about half of that, well, more than half comes from the organic matter. I won't get into it now, but I would estimate that the amount that comes from organic matter is about 8%, 8 milliequivalents. And so that about 6 or so would come from the clay, which if that's true, which I'm not saying it is, would make this about 30% clay. Well, I don't know if that's true or not, but that would be my guess based on the data. But you can play around with it. So what this is showing you is that yes, we need to add some nitrogen. This is for pasture, so that there was no nitrogen test done, but I would bet that's just being recommended. If it's intentionally grazed pasture where you have a lot of manure going on, you may not, but since the nitrogen test wasn't done, you don't know. You do need some modest amount of phosphorus. Again, that's P2O5, which you will not be putting on your soil. You'll be putting on another form of phosphorus, and they're suggesting 60 pounds of K2O, which would be around 50 pounds of K, actually. But when you're talking to the fertilizer people, you're talking in K2O and P2O5. Gotta keep that in mind. Oh, geez, I'm not going to get into this. This is too much. But this is, if you're talking about calcium, magnesium, and potassium, there are different systems to make the recommendation. And there are some people that are very enamored with the cation ratio system, which uses the ratio of calcium, magnesium, and magnesium for making recommendations. Has nothing to do with the nitrogen and phosphorus recommendations. Some labs blend both systems. We do it a little bit for magnesium, but this is getting into stuff that we just don't have time to get into. There's a whole lot of research. This was a summary of 100 years of research on the whole issue of the calcium-magnesium ratio. And basically what they came up with is that it's not that big a deal, that you can get into trouble using the cation ratio system if you're not careful. And I can show that to anyone who wants to see that. I better quit now so there'll be at least some time for some questions. I think I better do that. So I'm going to escape here. I'm going to quit here. And we got maybe 4 minutes.
Thank you very much, Fred. If anybody does have questions, please toss them in the chat. Especially even if we don't have time to answer them today, we will record them and make sure that they do get answered in future webinars.
Sorry that went so fast, but I didn't have an alternative.
No, I think we nailed the discussion questions throughout. And if we run a little long on that, that's okay. All right, I'm not seeing too much else. I'm seeing some thanks, Freds. But so I will share my screen as the wrap up, and then we'll make sure we close at 4.
Okay.
All right. So thanks again, everyone, for being with us today. And thanks to Fred for talking for nearly 2 hours straight. Much appreciated. I just wanted to let everyone know that next week on December 19th from 2 to 4 p.m., we'll be back here on Zoom. Our topic will be the living soil, and we will have two wonderful speakers, Wendy Sue Harper and Lisa Blazure. Actually, Lisa, I'm not entirely sure if that's how to pronounce your last name. I will make sure I nail that for next week. If you are all able to please fill out the webinar evaluation form, Mel just put that into the chat. There's space there to ask more questions. There's space there to tell us what you liked and what you didn't like. Once again, please do practice teaching and applying the information that you learned from today's webinar. I know there were many, many things. So hopefully one of them stuck and you'd be able to share that with a neighbor, a friend, or a producer that you work with. If you haven't yet, please set up your campus account. And then once you get there, you can go and read the homework for next week, which is chapter 4 of the Living Soil and Building Soils for Better Crops. Making sure to say that the right way this time. And you can also explore some of the recommended resources. The ones that are posted for this week too are some of the ones that Fred himself actually recommended. So please do take a look at those and take a look at the ones for next week as well. And if you do have questions or conversations, please enjoy the space in the discussion section on campus. I'm also just seeing there's a couple questions about how to get in contact with Fred in the chat. So please do contact Fred directly if you have any questions. If you think it could benefit the whole group, feel free to email us at info@franklincountynrcd.org, and we'll make sure that anything that gets answered, we can bring up into the next webinar. With that, thank you very much, everyone. And we are excited to see you next week. After next week, we'll go on a couple week break and then come back in January. So please do try to make next week live, and then we'll give you a break from all of the information and learning. But enjoy the snowy weather wherever you are, if you can enjoy it. And thank you once again, Fred, for everything.
My pleasure.
Thank you, Fred.
Thank you. Good seeing you.